Next up, we have Mark Emmelfarb from Dyadic International.
Well, thank everybody for coming. It's a big audience. We're excited to be here at BioProcess International after more than 2 years of being locked up with COVID. And I think that one of the things we'd like to highlight here is that our company is committed to make healthcare accessible and affordable, not just in America, but basically on a global basis. And we've developed a technology over the last 3 decades to make proteins for world health, whether it's feeding the world, fueling the world, or healing the world. It's our safe harbor statement. We're a public company on NASDAQ under DYAI. I would suggest you take a look at our journey and watch us as we go. So our mission here is to make proteins, manufacture proteins, do it quickly, do it cheaply, do it at large scale for feeding, fueling, and healing the world. We've already successfully fed and fueled the world for over 25 years. We made enzymes at large scale. When I say large scale, I mean taller than this building at 500,000 liter fermenters. And now the final frontier for us is to heal the world, and we're actually waiting anxiously for South Africa to potentially give us the green light to put a antigen produced from this very powerful, fast, rapid, highly productive cell line in a human being for the first time. And that's going to be like what Elon Musk did to the auto industry. Is people said, you can't do this. Well, we're going to do it, and we're doing it now, and we're going to do it over and over and over again. And it all starts really with our journey 30 years ago when we first found this C1 cell line, which is a filamentous fungal cell that doesn't sporulate. It's been through grass studies. People constantly have asked, well, have you been in humans? Have you been in humans? And the answer is, well, we've been in chickens, pigs, cattle, and mice, hamsters. But the answer is no, because we didn't have an opportunity to. And COVID opened up the door for us to actually do what for us is most important is safety. For the first time, putting a protein into a human being and demonstrating that what we already saw in the animal studies and the US FDA GRAS approval, where we didn't have to purify it at all and put it in animals, it was safe even without purification because we have no endotoxins, which is what bacteria has, E. coli. So we don't have to remove that because it's not there. We don't have any viruses, which are in baculovirus insect cells or in CHO cells. So the pedigree is actually safer. And so now we're going to move on and moving forward towards that goal. So it's about human health, it's about animal health, it's about alternative proteins. And each one of those areas are massive, large, multi-billion-dollar markets in the vaccine space. We can make more vaccines in a short amount of time at a lower cost than anybody on the planet. And we'll show you why and give you the data to back that up. Therapeutic proteins, that's still being developed with our glycoengineering for glycoproteins, but we have humanized now cells from this fungus, C1. And we can make things quicker, faster, and cheaper there as well. And then in proteins and enzymes for alternative proteins for things like cultured meat or cheese and things like people are looking at now for more sustainable long-term way to feed the world and heal the world. So in human health, you can see here some of the parties we're dealing with in the human health side, working towards that phase 1 clinical trial, working with Janssen on a couple different proteins. And these are proteins which are very difficult to produce, like what you'd call difficult to express proteins, potentially things like trispecifics or bispecifics that are sometimes CHO cells fall short, so they got to pick it up or trash it. And rather than just killing a program or trying 2 years to try to make it work, come to us and we'll help you make it work faster with higher probability because we can express things at higher levels. So if you take a hit from 20 grams per liter instead of 2, you're still in the ball game and you can move your platform forward. On animal health, we're working now with several parties, but Phibro is publicly one of the top 10 on 2 different programs there in poultry. And alternative proteins, one of the largest global food ingredient companies, is funding research for this technology to move forward in the cheese space. We work in stainless steel fermenters or single-use bioreactors, virtually with the same productivity and the same yield in the same time. So it's your choice. You can work in a stainless steel or use plastic bags. But the advantage here is that we've scaled up to 500,000 liters. So we're scaling down, we're not scaling up. As you know, biotech is fail to scale. So the valley of death, going from a test tube to a fermenter and scaling it, well, we're going 25 to 50 to 100 times down, not up. And it's run on glucose and simple chemically defined media, very low cost. So not only can we make more for less, it costs less to actually feed it, and we can scale it to virtually any size you want. pH 5 to 8, temperature range 20 to 40 degrees. Those are huge differences. CHO cell, if you move a half of the temperature degree, you kill it, program's done. So the more robust, more versatile. These are examples of yields. These are record-breaking yields. These are just the beginning. We haven't spent 35 years making human proteins. We spent 30 years making industrial enzymes. We're applying the power of that technology to what's clearly an inefficient world to us. And so if you think about it, we made these proteins without even doing fermentation optimization at levels that people just shake their head. And I'll get into a couple examples of that. But if you look at the antibodies at 24 grams per liter in 7 days, the average for CHO is somewhere around 4 to 6 grams per liter in 14 days. But that's just part of the story. It takes them about a month to get into a fermenter. It takes us a couple days. And then the CHO media is 20 or 50 times more expensive. So we can run 3, 4 batches in the same time and run one. We can do it with much lower media costs, higher yield, and we don't have any viruses. So you save 1 or 2 months on a downstream release doing the viral inactivation. So it seems to be a no-brainer, but we're stretching the industry's conservative nature and trying to show them that this is the future. Synthetic biology, faster cells, the doubling time, 2.5 hours versus 24 hours, low-cost media, more rapid, higher yield. The world is not getting medicines to 98% of the population on the planet. And they seem to be okay with it. I'm not. I think the people that we're working with get it. And as an industry, we have to do what Elon Musk did. And basically, there was a lot of naysayers saying, oh, he's never going to have it. He's going to run out of business. He's going broke. In the end, he's the richest man in the world, and people are going to electric engines. So if we really want to get healthcare to people affordably, guess what? We need to do it in a more efficient way. We need to transform the industry, not improve marginal improvements and patting ourselves on the back because we got 2% more last year than we got this year. We need 10 or 20 times more to do this in the right way. This is just some of the targeted areas that we're focusing on or with our own and with our partners. You can see here, I'm not going to go through the list. There's a variety of things. We've got a non-human primate study that we just completed on a COVID-19 antibody. And again, this is more about proving for the first time, in this case, an antibody in humans. The other one was an antigen in humans, of a platform that's going to revolutionize pharmaceutical biomanufacturing, just like we've done with Shell Oil, BASF, Abengoa Bioenergy, and DuPont. So DuPont didn't pay us $75 million to have rights to use this because it's not productive. That's in an industry where you're going to pump out massive volumes at very low cost at large scale. So bringing it into this industry is sort of like Tinker Toys in terms of scale. It's just adoption, it's showing safety, getting efficacy. But when we crack that egg, it's going to really make a difference in healthcare and lives on a global basis. So we have all the molecular tools you could ever want to do gene knock-ins, to do gene knockouts, to make pathways, to do metabolites, therapeutic proteins, viral antigens, vaccines, whatever you want. You can see that the cell line, we changed the morphology, the way the cell grows. It doesn't grow in a morphological, high viscous solution. It grows under very low viscosity. At the end of the fermenter, it's around 10 centipoise instead of 10,000. And that's why we can scale. It's feed uniformly, aerate it uniformly. It's a great thing. We can program it with virtually anything you want to do. We can do site-specific integration. put it 1 or 2 gene copies in one site, 1 or 2 gene copies in another site. We're developing technology to what we call hotspots or loci, which is exactly we know from where this protein will be expressed at a high level, because we spent hundreds of millions of dollars together with us in Shell and BSF and Abengoa and others in the industrial world. We know where to put these genes to get high productivity. And we're targeting them directly. And from a regulatory perspective, that allows us to do things over and over and change, like kind of seasonal flu program. So for COVID, for example, we can put in Wuhan, pop it out, put in alpha, gamma, delta, Omicron 2, Omicron 4, 5. You'll see we made all those very quickly, very stably, and we can produce massive amounts of those proteins. So the platform here, you can also add secretion factors, folding factors, and take it even higher. So even as good as the results are now, and as you're hearing today, it's going to get better, quicker, faster, and cheaper. Recently, we went from 2 grams per liter to 11 to 22, all within 3 months, by applying different technologies and approaches genetically to the cells. We moved from industrial to pharma. We had to remove the background of the proteases, because if we're trying to make protease or proteins, proteases chew them down. So we knocked out, in this case, 15 different protease genes. The cells are still healthy. They still produce large volumes. They do it stably. And you'll see some examples here on the right of a monoclonal antibody expressed at very high levels, very stably. We then had to change the glycosylation structure so it's human-like or CHO-like. For monoclonal antibodies, the belief is you need to look like a CHO cell. And that's because for 35 years, they've been using CHO cells. Well, actually, we're actually more human. So we're probably can make biobetters, or we can make new molecules with human-like glycosylation, not hamsters. So we theorize that might be a little better. But also from a vaccine perspective, you don't want human glycosylation. You want sort of off-human to create more immunogenic protection, better response. So you'll see that not only do we make safe, effective, protective vaccines for human and animal health, but we're doing it at levels, just remarkable levels, and we're seeing great results on that regards. This is the example of nivolumab. For those of you that don't know nivolumab, it's Opdivo from BMS. It's their $8 billion drug. The average of a protein is 4 to 6 grams per liter in 14 days. It takes them a month to get it into the fermenter. Well, here we are in half the time at 22 grams per liter. So that would be like 44 grams per liter in the same time versus 4 to 6. There's your 10-fold number. On the upstream side, you can see it's stable. And we're now developing the N-glycan or human glycan cell lines to produce that as well. This is an example of infectious disease antibodies. And here, I just want to take a minute, because I think the world is not looking in the right lenses when it comes to infectious disease antibodies. Because you take out a CHO cell and you're not repeatedly dosing people over and over again like Humira for rheumatoid arthritis. You're going in once, you're fixing the problem, and you're done. So you want to use the most productive cell line. If they bind and neutralize and solve the problem, and you can make 5 or 10 times more of it, that means we can treat and save 5 or 10 times more lives by choosing to supplement CHO. So in the US during this pandemic, in Delta, if it was more virulent, we were in deep trouble. We had shortages as it was. If you lived outside the USA, 98% of the world didn't get these antibodies. They either weren't accessible at any cost, but even worse, they weren't affordable for most of the world anyways. And so we were working with and talking with a few weeks ago with WHO, and we've demonstrated now in a non-human primate study that using the human-like original cell line, it's 5 times more productive than the glycoengineered cell line. It binds, it neutralizes the virus, and there's no side effects. So putting them in CHO cells is kind of like overkill for an infectious disease when it's a one-time treatment. Here's an example of, on the left, a COVID-19 antibody. It's not our antibody. It was done in conjunction with European scientists we worked with. But you can see all the way through Omicron 1 and 2, you didn't have to reduce or increase the dose because it worked effectively on all of them. So we made it obviously in C1. You can make 3 or 4 batches in the same time as CHO, but it actually worked better. So again, we're agnostic. We're taking other people's genes in most cases. You'll see where we're advancing our own programs as well. And you see where Regeneron failed completely with Omicron, suffered with gamma and beta, where there they had a higher dose, but in Omicron didn't work at all. There, you had to use 10 times more of it for Omicron. So the solution here is really we need to have more efficient, better ways of doing things that are quicker. We can generate cell lines 2 to 3 months faster, make more batches, higher yield, lower cost. So you see here the lower cost, faster production, higher yields. You know, we need to rethink the way we're doing things. We can't keep doing the same thing over and over again, expecting different results. Obviously, Einstein and others define that as insanity, but we keep doing it. And you guys need to help make that change. Because if you don't do it, people are gonna die or they're gonna suffer, they have more pain and suffering unnecessarily. And as a human being, that's not acceptable to me, and I don't think it is to you as well. So now the other thing is when you talk about antigens and vaccines or whether it's an antibody, we're agnostic how you want to use it. You want to use an intranasal vaccine? We're making the protein. You can apply it any way you want. We're making it in high levels, we're making high purity. So now they're talking about let's make intranasal vaccines, right? Maybe they're going to last longer, give better protection, mucosal immunity, whether it's intramuscular, intranasal, intradermal, oral. In our case, we're just making the protein, we're making it high purity. In fact, our own COVID-19 vaccine was over 99% pure. If you're over 90% as a vaccine, that's pretty good. So we're way over that. And here's an example that you should sit back and take notice and be appalled by what happened, because we spent 5 years in the Zoonosis Anticipation Preparative Initiative in Europe called ZAPI in 2015 to 2020, preparing for a pandemic. God forbid we have one. Is there a way to make vaccines and antibodies quicker, faster, cheaper, at large scale with surge capacity? And in this program, we were chosen our cell line because it made 300 times more than baculovirus insect cells. It did so in 3 days less time. You have no viruses to remove. It's a no-brainer. So if you look at that and you think about it, and you look at companies like Sanofi who used insect cells or baculovirus, or Novavax, 300 times more, 3 days less, no viruses to take out. It would have got there quicker. You could have given it to more people for a lot less money. And we're working on that in South Africa right now. And we're working on that also in India. The Indian government has provided funding to one of our licensees to move a vaccine into human beings for phase 1 and phase 2. In this case, it was an animal study, Schmallenberg virus, Rift Valley fever virus, safe, protective, effective, more efficient, more affordable. And these are the studies that are on our website, cattle studies, the mice trials, lamb studies, again, which I mentioned earlier. We've been in sheep, cattle, mice, hamsters. Apparently I forgot about the lambs, the rabbits. So we'll see how we do in humans. Hopefully we'll get in there soon. And I'm pretty confident that we won't see any side effects or any negative effects, and all positive news going forward. Again, this is a proof of concept of our COVID vaccine. As I mentioned, within 2 months or less, we had stable cell lines. So by June or July of 2020, we were ready to go. You can see here on the study as we get into it, the purified here was at about 1 gram per liter at over 90% purity. That could have gone into a human being right there, but then it takes time to go through all the FDA or EMA or different regulatory agencies to bring it forward. But this was stable, it was highly produced, it was very pure. The mice studies, we did a BALB-C mice study, which ended up very positive. We did a ACE2 transgenic mice study, which is the mice either live or die, so it's black or white. Or hamster studies, it's kind of gray. How well did it work? Where did it work? These things either live or die. And guess what? We did a 2-shot study and a 3-shot study, 6 months or more ahead of the world thinking you need boosters and demonstrated that the third shot gave even better protection. This is our tox study. This tox study was done, it didn't just demonstrate safety. If you look at the cells on the right, in the recovery period, the mRNA vaccines are gone. We're still producing antibodies in the B cells. So we have potentially better persistence. So potentially a better, safer, cheaper, larger way to make vaccines. This is now in regulatory, just kind of, again, we're investing in, we want to have demonstrations now in phase 1 with the COVID-19 antigen, vaccine DY100. We're just waiting, hopefully very soon, to get news to go forward. And then, of course, we have the non-human primate study on the antibodies. So I think this gives us, and then, of course, the tox study and the graft study, good support and feelings for the safety. In fact, the pedigree of a filamentous fungi, just like yeast, there are no endotoxins, there are no viruses. The pedigree is actually better than something that makes endotoxins you got to get rid of. It's actually better than CHO cells or insect cells that have viruses you got to get rid of. So you would think that this would be something that people would be running at the door rather than running away from the door to use. But again, doing the same thing over and over again, expecting different results, is what Big Pharma has done for generations. And we need to make that change. Together, we need to do that. So here's just an example at 2 grams per liter with the recovery rates of 70% of fermenter capacity, recovering 60%. You could have made 1 billion doses or 2 billion doses in a week in a 62,000 liter fermenter. Just think about that. That's almost 3 times what the US needed could be produced in a fermenter in a week if we would just plan ahead. And we did plan ahead with the ZAPI program. We were involved with that. Unfortunately, Europeans never funded it and left it on the shelf, as did Warp Speed and others. So here's some other things here again. Guess what? Here's an example. We actually made alpha, beta, gamma all in the same cell at the same time in the same fermenter. So that was a trivalent. And that was done over a year ago. Something new. You might have heard about pan-coronavirus, better protection, longer protection. How do we do it? Well, you have people like Walter Reed has this ferritin nanoparticle vaccine. They're in phase 1, phase 2, showing longer, better protection. Duke. Well, guess what? We made the Wuhan here at 3.5 grams per liter. That's a massive amount of protein. And that's a ferritin nanoparticle protein. We've now made it also in Delta. We've made it in Omicron. And so here's just an example of some of the things we made for influenza. We've got H1N1, we've got H5, H7, we've made neuraminidase. So we can put together a mix of a very, very protective, probably better seasonal flu vaccine protection, which we all know now we need. And we should be targeting for better flu vaccines, not accepting what they've been giving us for decades. So we can blend these together. You can see here, we have all those different cell lines. We can do it for the antibodies. We can do it for the antigens for the vaccines. And so here's an example of seasonal flu. This was H1N1 with Oslo University versus Sanofi's flu block on the bottom. So with 1 hemagglutinin and a ASO3 adjuvant, we were 100% survival in a challenge study. And even without the adjuvant, we were 70% of the mice survived, which from recombinant proteins is unheard of. So we're getting this extra protection, immunogenicity from that glyco pattern is what we talked about earlier. This is from Sanofi Pasteur themselves, comparing this to baculovirus insect cells. 1/3 the amount of protein, better results, made 100 times cheaper. Seems like a no-brainer. And this is not our slide, it's our slide deck now, but this is on our website, dyadic.com, Media Center, scientific presentations, and you'll see it on Sanofi slides. So this is done by independent third party, largest seasonal flu company in the world, but yet they're using insect cells. This is an example of neuraminidase, which is very difficult to express. And this is both IgG titers and the ELA assay. 800 milligrams per liter before we even optimize it. That's a massive amount of antigen. And you can see down below the expression levels for H1 H5, H7, and the neuraminidase. So these are all things we've made now in our technology with C1. Toxicology publication on our website. You can see the other vaccine publications all on our website. Get into the details. There's more publications coming. This is the right time. Actually, it was the right time at the right time and right place, but it still is the right time, at the right platform, at the right place for several reasons, not just for pandemics, but bringing low-cost, affordable healthcare to human beings on the planet everywhere. And we're working diligently with low and middle-income countries that are adopting this technology in Africa. They're adopting the technology in India. We're working with Janssen here for the West and a number of other companies as well. And I want to thank you because, again, a vaccine or drug that's unavailable or unaffordable is 100% ineffective. So together, let's transform